Hey guys, hope you're doing well. Today we'll look deeper into genetics, focus on allelic animals such as blue-eye leucistic, and one of my favorites, the candino. So get ready, let's dive in. BEL, or Bell, which stands for blue-eye leucistic, was considered the holy grail of ball pythons, and now we know how to produce them. There are actually several combinations of genes that can produce the Bell. The genes in the Bell complex are butter, lesser, Mojave, Phantom, Russo, Mocha, Mystic, and Special. Most of these genes, when combined, will produce a bell. For example, Mojave to Lesser, Butter to Russo. You can also achieve a bell with two copies of the same gene, such as a Russo to Russo, or a Lesser and a Lesser and a Mojave and a Mojave. Now, because there are so many genes in the bell complex, let's keep things simple and look at another allelic animal, which is one of my favorite, a candino. Candino consists of two genes, the candy gene and the albino gene. These two genes are both recessive, and we know recessive gene had no visible effect to the animal when the animal only has one copy. So you think about it, it has one albino gene, which means it would look completely normal, and it has one candy gene, which again have no visual effects, so it'll look normal. But in reality, both the albino and candy gene, when coming together, actually makes an animal look completely different than an albino or a candy or a normal. So to understand how this happens, let's take a closer look at DNA. A strand of DNA looks like a ribbon that's twisted. It's in a shape called a double helix. Let me try to draw that. Let's imagine this is a strand of DNA from a snake. And as you can see, the strand of DNA consists of pairs of genes. So each one of these little dashes is a gene, and they come in pairs. Half of it is from the father and half of it is from the mother. Now let's try to untwist this ribbon of DNA so it's easier to see. Now we can see the DNA as a flat ribbon, and it kind of looks like a ladder with rungs. Each rung is a locus, and at each locus there is a pair of genes. The genes come in pairs again, like always. Half of it's from the father, so one is from the father and one is from the mother. When the snake needs to produce sperm or egg, it goes through a process called meiosis. That's the process where DNA splits in half, and each half either becomes a sperm or an egg. So in the case of a male, these would both be sperms, and they would consist of half of the DNA of the original, or in the case of a female, both of these would become follicles. So let's assume we have our male here, and its DNA went through meiosis, so we have two sperms. And over here, let's assume we have a female DNA, and it went through meiosis and produced two follicles. So if we took half of the male DNA, which is a sperm, and half of the female DNA, which is a follicle, we pretty much get back at fertilization a whole strand of DNA. And this whole strand of DNA is the foundation for a new baby ball python. So depending on the genes that are from this half of the DNA and the genes from this half of the DNA, they come together to produce this DNA, and depending on those sets of genes, that's what the snake would be. So let's, for example, pretend that this gene right here is a pastel, and this gene up here is a spider. So what we have here is a pastel spider baby ball python. What's important to note in this situation is that the pastel and the spider are located at different locus on the DNA strand. So how is this related to allelic animals? In our case, let's use candino. So going back to the original picture that I drew, let's pretend that at 1, 2, 3, 4, 5, at the fifth locus point, this is where the albino gene would reside. So in this case, if we have one albino gene, this animal would be a het albino. But what's funny is that the candy gene also resides at the 5th locus point. So in theory, or in actuality, this animal is het albino and het candy. But because they reside at the same locus point, these two genes interact and produce a candino. Now, this visually changes the appearance of the snake, even though the snake does not have two copies of the gene. They have one copy of the albino, which is a het albino, and in the case of a het albino, it should not have any visual change. Also, the candy gene is also a recessive gene, so it requires two copies of the candy gene in order to visually be different from a normal, but because in our case it is a single copy of the candy gene combined with a copy of the albino gene living at the same locus point, it produces an animal that is visually different from a normal. Now, to kind of make things interesting, let's assume that this DNA here, this strand of DNA, only consists of a single albino gene. The rest of it is normal. So in theory, this animal, which is our male, would look completely normal. This would be a het albino male. That's it. Just keep things consistent. 1, 2, 3, 4, 5. This would be the albino gene. 1, 2, 3, 4, 5. This would be the albino gene. So this male has one copy of the albino gene, and when it went through meiosis, this half became a sperm and this half became a sperm. So we're interested in this half, this sperm right here. Now over here we have the female. Let's assume that she is a het candy. So at 1, 2, 3, 4, 5. At the 5th locus point, she has a candy gene. So really she would be a het candy. So over here we have a pastel het albino male. Over here we have a spider het candy female. And this sperm with the albino and pastel gene, and this follicle with the candy and spider gene came together through fertilization. We have 1, 2, 3, 4, 5. At the 5th locus point, we now have an albino from the sperm and a candy from the egg. So now we have a candino, a pastel spider candino. This would be our allelic animal. The candino is an allelic animal because the albino and the candy gene falls on the same locus point and they interact with each other. Now, just to keep things simple, let's remove the pastel and the spider gene from the equation. We'll just look at the albino and the candy gene. So in our case, let's use the Punnett square. We had a albino normal male to a candy normal female. Create the Punnett square. Again, we'll put the male on top, which is AN, and the female off to the left. Working out the Punnett square, we combine the albino and the candino gene. Albino and candy gene produces the candy. This is a het candy. This is a het albino, and we have a completely normal animal. So in this pairing, we have 1 out of 4 odds of producing a candino, and we have 3 out of 4 odds of producing normal, visually normal, but we know, 1 out of 4 would be a het candy and 1 out of 4 would be a het albino. But what if we wanted to produce a whole clutch of candinos? Let's use the Punnett square and work backwards. So we know that if we want the entire clutch to be candino, all of these quadrants would consist of one albino gene and one candy gene. So let's isolate to the first quadrant here. We have an A and a C. So we know that we need an A or a C, and it doesn't really matter where you put the A and where you put the C. So we'll just pretend we have the A here and the C here. So that will produce this quadrant here, A and C, right? The A and the C combine to produce AC. And now we have this quadrant down here. So we already have an A up here, so that's where this A comes from. Now we need to make sure that we have a C, so we'll put a C here. Moving over to this quadrant, also an AC. The C comes here. That's how that C arrives. Of course, we'll make sure we need an A. And then lastly, just to confirm this last quadrant, A and a C combine to be A and a C. So we know that in order to produce an entire clutch of candinos, we do not use any candinos as the parents. What we need to do is use an albino paired to a candy. A visual albino to a visual candy will produce an entire clutch of candinos. So to sum it up, an allelic animal has two genes that are different, but they exist at the same locus point, and those two genes interact with each other as if it was a homozygous pair of genes. The candino belongs in the recessive complex. As I've mentioned, it consists of a candy and an albino gene. There is another gene in this complex, which is the toffee. A toffee and an albino gene produces a toffino. There is also the black-eye leucistic complex. One of them consists of fire and sulfur, so breeding a fire to a fire or a sulfur to a sulfur or a fire to a sulfur will produce an animal that is all white with black eyes. Also, there is a yellow belly complex. In this complex, a yellow belly and a specter produces an animal known as a superstripe. So that's one example. And lastly is the cine complex. So an animal with two cinnamon genes or two black pastel genes or one cinnamon and one black pastel gene will produce an all black snake. However, these are lethal genes, so just be careful because it may result in an animal with physical or neurological disorders. So that's it for now. If you find this video helpful, give it a like. If you have questions or ideas for more videos related to ball python genetics, please comment below. If you want to be notified of future videos, hit the subscribe button and I'll see you next time.